Okay. We left off last week talking about motor neurons. Okay, talking about motor neurons. So we saw the different structures. We talked about the cell body or the soma. We talked about dendrites. We talked about the axon. I want to point out a couple of really, really important structures in the cell body. Okay. We have what is called the nucleus. You guys know what a nucleus is, but within all of this, sort of in our nucleus, we have a thing called a nucleolus, which is very sort of aptly named. But the part of all of this that's important is that it has what's called ribosomal RNA in it. And this, the nucleolus in the portion of the nucleus here, is what drives the ability of the neuron to make proteins. Okay. We don't always think about it this way. We don't always think about it this way, but your neurons need a lot of protein. They need a lot of neurotransmitters. They have to make a bunch of different things, okay, in order to stay healthy. They have to make things in your spinal cord and carry them all the way down the axon to the neuromuscular junction. Feet, okay? Very, very long distance. The other problem about the cell body and the nucleus here that's something that's unique to neurons is they do not have an ability, okay, to divide. They do not have an ability to replicate themselves as we move along. And so because of that, if something happens to a neuron and it gets damaged, we have a little bit of an ability to repair. But if something happens and that neuron dies, it's gone. Okay? It's gone. It's never coming back. That can be a real problem. Okay? And so you are born with however many neurons you're going to have. If we damage them, if we lose them, then we can begin to lose function, and we'll note some of that stuff as we go along. Okay? Now, here we have these little structures kind of nearing. So the area right here where the axon comes off of the soma is called the axon hillock. You don't have to know that. But these little structures here are what we call Nissl bodies. Again, they're going to work with the nucleus and the nucleolus. They have very, very heavy amounts of ribosomes and endoplasmic reticulum. So they're going to help us make proteins, make things here, package them up, put them into vesicles, so we can then transmit them down the axon, move them down the axon to the neuromuscular junction. Okay? The way that we move them down is through what's called cytoskeletal proteins, or a cytoskeletal protein network. You guys may have talked about this in your intro to bio class or something, or if you've had cell bio, there are these little things called microtubules, microfilaments, okay? And so microtubules are kind of like actin, okay? They're a slightly different version, it's the same general idea. And there are proteins, not myosin, but proteins that function like myosin that walk down these microtubules, okay? And so what you do is we make things here in the cell body, we package them up with the Nissl bodies, we package them up here, and then we hook these little vesicles onto these molecular motors. They're called dynin and kinesin, which you guys don't have to know. And then they walk along these microtubules all the way down the axon to the neuromuscular junction. Okay. It takes a long time and a lot of energy to do this. So it's a really, really important process. And this will become apparent when we see how we try to recycle things at the neuromuscular junction so that we don't have to make any extra things here. But any disruption to this can cause impairments in function. Okay? Any disruption can cause impairments in function. And that's going to be an important thing as you age, as you accumulate damage to these sort of neurons or things like that. If you get any kind of blunt trauma on them, that can also be a big deal. Okay. All right. So there's not a lot to show. Well, if it would actually go forward, right? This is more of a microscopic view of what the dendrite network looks like, okay? The cell body is here, and you've got axon that's coming out like this, but sort of everything else is going to be dendrite. So I know that in the one picture, they've got these little small, kind of very few things that are going on, but it's actually this very, very dense kind of web-like network of things that are going to pick up signals from all the way around. So that's kind of a cool thing. You don't have to draw a picture of it. I just want to show it because I know some of you guys, it helps to visualize these things. Okay. So two more concepts to try to wrap our head around with the location of motor neurons. Okay. So the motor neurons are only going to be contained in certain very specific places in the spinal cord. Okay. I will illustrate a few of those here. What I want you guys to know is I'm not going to ask you, hey, are the biceps at C5 or C6? I'm not going to ask you guys that. This is just in a very general sense, do you understand where they are? Because where they're located is going to play some level. The reason they're where they are is going to come back and help us understand function. Okay. So in general, okay, we have upper body motor neurons in our cervical vertebrae. Okay, they're going to start, weirdly enough, okay, the diaphragm, which is probably the lowest, right, kind of location wise, from sort of cervical vertebrae, it's the most distal, is contained at the highest, or innervation at the highest level in the cervical vertebrae. Okay, it's usually about C3, C4. Why would it be important? If we get closer up here to the skull, where there's likely more protection, why would we want the diaphragm to be the highest? We need to be able to breathe. That's right. Very good. Okay, we need to breathe. So if all else doesn't work, breathing keeps us alive. So diaphragm is up there. And then as we move sort of down the spinal column, it's going to generally be sort of proximal to distal with what you're thinking of, right? Shoulders, wrist extensors, triceps, hands, those kinds of things. Okay, we're going to move our way down as we're going to go. In the thoracic vertebrae, we have much fewer motor neurons. There are some, but we have fewer in the thoracic vertebrae. Internal, external intercostals, so those kind of accessory ventilation muscles are going to be there. Lower thoracic, right, abdominal muscles, those kinds of things is kind of lined up about with where we are in the spinal column. Then once we get down into the lumbar and into the sacral vertebrae, lower body muscles. And it again generally follows kind of proximal to distal, hip flexor, knee extensors, dorsiflexors, right? The toe extensors, and you get way down into sacral, and you've got a little bit of stuff that's going to innervate some of those, that smooth muscle and stuff around the bowel of the bladder, those kinds of things. Okay? And so for the most part then, the vast majority of our skeletal muscle is going to have innervations in the cervical vertebrae and in the lumbar vertebrae. Not a lot sort of in the middle. Okay? This is important because of function. Okay. The other part of this, let me show you guys the next slide, and then we'll talk about it collectively, is this. Okay. Here we are at L4, and you're kind of seeing through the vertebrae down to about S2. So S1 is here, L5 is here. And what you guys can see is we've zoomed in on this area, right, of the dorsal horn of the spinal cord. And what we're trying to illustrate, these little tubes in here are trying to show that at that vertebral location, all of the motor neurons, all the cell bodies of the quadriceps are in roughly the same location, in roughly the same place in the spinal column. They're all kind of sitting here together, right, at L4. And then as we go a little bit more distal, we've got the TA, you've got the plantar flexors, okay, we've got glutes that are down here, which are a little bit weird that they're a little bit lower, but that's what you're going to see. If we were to look up in the cervical vertebrae, we'd see the same thing, which is for our upper body muscles. So the two things to take away, okay? There is a distinct anatomical location in the vertebral column where motor neurons are going to sit, okay? And at that location, all the motor neurons, every one of them for a given muscle is going to exist in about the same location altogether, okay? This creates several kind of functional consequences for us, okay? Why would it potentially be beneficial to have all of the motor neurons in the same place, right? All of my quad motor neurons are in exactly the same place. All of my bicep motor neurons are in exactly the same place. What might be a benefit? What do you guys think?
Less area to stimulate the muscle.
Less area to have to stimulate. Okay, good.
Maybe a little bit faster, like motor unit recruitment.
Faster recruitment. Okay. How might it be slower if it wasn't this? It's an interesting concept. What would happen? Let's think about it in the opposite end. What would happen if I had some quads here, some quads here, some quads way down, let's say like S3. And I get in a situation where I'm doing a 1 rep max squat, I'm way down, okay? It's super heavy. I gotta turn them all on at the same time, which we haven't even really talked about. We're gonna get them all turned on, but they're all in these weird different places in the spinal column. Does that seem like a good idea? Probably not, okay? And so the reason that we're gonna do this, we'll talk about the bad part of it in a second, is it's exactly what the guys up here had said. By having them all in the same place, we know where to send our focused impulses, okay? And it gives us an opportunity to turn as many of them on as we need at roughly an identical time, okay? So if you think of the nervous system, right, as this long extended sort of wire, and as things move from the brain down, right, it's gonna, an action potential from the motor cortex will pass through the cervical vertebrae first, then there'll be some time lag, then it will go through the thoracic vertebrae, time lag, lumbar vertebrae, time lag, sacral vertebrae. Then there's even more lag once it gets down there to get actually out to the muscle. So if you had some motor neurons in the cervical vertebrae, they're going to get an action potential first, they're going to start to fire. Then it has to travel through the column, down someplace else, maybe to, let's just say, lumbar, fire additional ones, and then that all has to travel. So what happens then is, I'm doing my squat, and these are only, milliseconds, but I turn some on, and I maybe need more, but I can't get them right now because of this time lag, because they would all be in different places, okay? And so the advantage of this, they're all right here, is that all of them on right now, if I need them all on right now, or if I need to shut them all off right now, remove the thing, they're all off, okay? That's the advantage. That's the advantage, okay? What's the downside?
Well, if something were to happen to like just that single area, then you're kind of lost all motor function.
Right. Exactly. Okay. It creates a scenario whereby if there's damage to the spinal cord, or there's damage to kind of the neurons in that one area, that we may lose all of them. Okay. We may lose them all. So it's especially prominent if you have a complete spinal cord injury. So the cord gets completely cut, and we have no ability to transmit impulses to everything distal to where the thing is. And so even though the motor neurons are intact, and indeed that's what we know. We know that people that have a spinal cord injury, their motor neurons are usually fine. The ones below the level of injury still work. We just can't connect the signal to get to them. Okay. So you lose all function distal to wherever the damage is, or if you get some kind of blunt trauma, even if it's not a complete cord cut, you may damage like an entire muscles group of motor neurons, and then they're gone. Okay. Whereas if they were interspersed all throughout, you might have a weakened muscle, but you still might have some ability to be able to get some level of movement out of those things. So that's the trade-off. Okay, that's the trade-off.
This also applies to like damage from the stroke.
So the stroke is a little bit different because generally the damage from the stroke is actually in the motor cortex, right? It's in the brain itself, but it's the same general idea that there's going to be an area, whatever area that is affected or that loses blood flow, those neurons will die there. And so in general, the areas in the motor cortex, the neurons that are going to start the impulses that come down to the alpha motor neurons are all going to be in roughly the same place. And so you get damage there, and then boom, now I don't have use of that arm. And right, because of the hemispheric nature of the brain, if it's on one side, then you kind of can lose stuff all, it's sort of, you lose it bilaterally, you lose it on one side, but not on the other. So it's a similar concept, but the location of the damage is a little bit different. Okay. But again, this is the idea that when you have a stroke, everything downstream is still fine. So if you can get a little bit of function back, you can relearn some things back in the motor cortex, all the peripheral structures are still going to be relatively intact. Okay. Cool. Okay, so now, anybody, y'all are maybe so young that you've never had this problem. I have this problem pretty much right now, every day. Anybody ever hurt your back? Okay? Yes? Do you hurt your back and you get like weird, like shooting pain or anything? Or do you hurt your back and you feel like, ooh, my muscles feel a little bit weak in those ways, like I cannot make use of everything. Okay? We don't talk about sort of back injuries in this class, it's kind of beyond our scope, but I will show you guys some things. The reason that low back injuries, lumbar back injuries, and these kinds of things can be so devastating, all comes back to how structure in the spinal cord and where nerves are going to enter and leave the spinal cord and your vertebrae impart this function. Okay? Here you can see, okay, this is the dorsal horn, this is the ventral horn. So this is the side that faces your back, this is the side that faces in. We've got one vertebrae here, okay? So what you can see on the dorsal side, okay, these are going to be, we've got dorsal, we've got ventral. On the ventral side here, here's where your motor neurons are going to move out. On the dorsal side, you've got afferent information coming back from the periphery, coming back in, and they're all going to kind of co-locate right here in the spinal column. But where these things leave, right, where all of this bundle of axons is going to leave the spinal cord, it's all right together. And it leaves through a little hole essentially around those bones around your vertebrae called the foraminal space. And so if you do something and you pinch this, or you pinch up here, if you pinch on the afferent side, boy, it hurts. Okay. You get a herniated disc, and you squish stuff out, you squish oh my God, I cannot think this morning. Pads, what the hell are the pads? Cartilage, God. You squish cartilage out into the midst of all of this, and it presses on them and you can lose things. Or if you get stuff here, then it messes up how these nerves where they leave are going to be able to actually transmit impulses and all of that. Okay. But different muscles receive sensory information and send out their motor neurons at those very specific locations in your vertebral column. Okay. And that is why these are sometimes called dermatomes and things, but this is why you can figure out, like you can diagnose where a person has the problem in their spinal cord, in their back or in their neck, based upon what muscle is weak, what muscle is hurting, or what area of the body is hurting. Okay. All because of the way those things are going to be set up. Okay. from smashing my head into people in junior high and high school playing football, I have bone spurs at C4, C5, and C6 in my neck. And y'all will all at some point have this fun experience when you get older. You'll just sleep funny, right? And you'll do something, and I will occasionally, I will sort of sleep weird and I will pinch those things. I have permanent numbness from C5 that runs across my shoulder, down the front of my bicep, right down here into my thumb. And my other fingers are fine. But like the tip of my thumb at times feels like, you ever have like dirt on your fingers and you feel like you can't quite get that kind of feeling like you want it, kind of feels kind of weird and dull. That's what the tip of my thumb always feels like. It's from pinching a nerve. It's just never ever recovered from all of those things. All right. I don't really have strength loss. I did at one point, yay, rehab, on a bunch of things. And it's because I have bone spurs and I squish things out on both sides here. So this is how and why these things are gonna operate in that way. Okay, we talked about the motor axon the other day. We talked about nodes of Ranvier, talked about myelin. The cells that make myelin are called Schwann cells. Those are these purple cells here. Okay. The myelin is going to wrap up around all of the axon, and it helps us to increase conduction velocity. Okay. This process is called saltatory conduction. We're not going to get into sort of all of the mechanics of that. Just want you guys to know, as we will see when we make action potentials, that there are sodium and potassium channels that help us generate the action potential, that help us repolarize. And because of the sort of way the axon is put together, around where the Schwann cells and the myelin is, underneath them, there's no sodium or very, very low levels of sodium potassium channels. All of the sodium potassium channels are going to exist at the nodes of Ranvier, where there's not myelin. And that is going to essentially, this is not what it does, but it essentially allows action potential to jump from node to node and increase our conduction velocity. Okay. So that's what we've got that's going on here. The big thing for you guys to know, very, very simple. I have myelin, it increases conduction velocity. If you don't have myelin on your axons, you touch the hot plate and you can't pull your hand away fast enough. Okay, it's going to dramatically slow things down. Okay, so that's what's going to happen there. Okay. So the next thing that we're going to talk about is, you guys see, we're kind of moving our way out. We've gone from dendrites and the cell body and the spinal cord, the axon coming out of the spinal cord. We're heading down towards a muscle. Okay. And so I just love this picture because you can see the striations in an individual muscle fiber here and there. And then these are going to be axons. And then this area right here at the end of the axon, where it comes into close contact with the muscle fiber, we normally now call it the neuromuscular junction. It used to be called the motor end plate. Okay. You can make an argument that the whole area is the motor end plate, and then each little place where all of these little bulb structures are, where they come into close contact, each one of those, there's many, many neuromuscular junctions at the motor end plate. But the point is, right, that you will know, look at this, see, we've got a separate axon here, we've got adjacent muscle fibers being connected by different axons. Okay. And so we're going to come down. I'm going to show you guys some pictures here. The way the neuromuscular junction looks, its individual anatomical structures exist for a reason. And that reason is going to help us turn force on quickly, get fibers to fire quickly, and turn them off quickly and efficiently as well. Okay. So we look at this from a cartoon standpoint, okay? This is what I want you guys to know. I essentially want you guys to be able to recreate this picture right here. Okay? So things to keep in mind. As the axon comes down, and gets into very close contact with the muscle fiber, then there's going to be a large number, I say large, I mean, there's going to be 10s to 20 of these sort of bulb kind of bulges in the end of the axon. Okay. That's what this, you can see it comes in, it's narrow and it bulges out. Each one of those structures where it bulges out is sometimes referred to as a bouton. Okay, a bouton or a bulb-like structure. The reason that it has this bulb is going to be important in a second. Okay, inside the end, inside that bouton on the nervous system end, there are going to be a tremendous number of vesicles filled with a substance called acetylcholine. Okay, called acetylcholine. Acetylcholine is a neurotransmitter. It's a neurotransmitter. And so what's going to happen is an action potential comes down the axon. It's going to reach the bouton. And there are specialized receptors that are going to exist sort of up here in the more proximal end of it. And those receptors are what we call voltage-gated calcium channels. Voltage-gated means that when the action potential comes through, it opens them up. That lets calcium move into the bouton, and that calcium coming in is going to signal for these vesicles with acetylcholine to dump the acetylcholine out here, dump it out into this sort of area so that it can float around in close proximity to the sarcolemma. Okay. Nervous system side, muscle side, then we're going to talk about what's going on the muscle side in just a second. Okay. These are our vesicles. Vesicles actually integrate into the membrane, so their vesicles are a lipid bilayer just like this. They integrate in and they dump the acetylcholine out into here. The space between the nervous system side and the sarcolemma, okay, is going to have a name. And that name is going to be called the primary and the secondary neuromuscular cleft. Okay, cleft. So at the cleft on the muscle side, you will note there are folds. Okay, there are folds in the sarcolemma here. Most of the sarcolemma is flat, it's straight, it does not have folds. But under, at the junction point, there's going to be these folds. Okay. The primary cleft is this space right here. Okay. And then the secondary cleft is the space down inside the folds. We fold the membrane for the same reason that we bulb out on the nervous system side. It all has to do with surface area. Okay. It all has to do with surface area. We have more surface area on the nervous system side so that we can hold more acetylcholine. We've got more area to be able to release it from. And then on the muscle side, on the sarcolemmal side, we fold because it increases surface area. And you will note, both in the primary and in the secondary cleft, we have these little green structures which are denoting receptors for acetylcholine. So we have more space to release acetylcholine, and we have more space to house the receptors for acetylcholine. What this does is it makes for very, very, very efficient transmission. We don't have to release a huge number of acetylcholine, okay, because we've got lots of receptors because there's lots of surface area on the muscle side, okay? So we can get transmission, we can get the muscle to turn on without having to release all of our acetylcholine and sort of hope that it works. But we also, because there's lots of area, if we need to really, really turn everything on, we can dump a whole bunch and be confident that we're going to get good transmission there. Okay. We're maximizing the release, and as I'm calling it here, capture. The surface area maximizes our ability to release it and to bind it to its receptors. Okay. Again, anatomy informs physiology. Structure informs function. Okay. All right, so we talked about these sorts of things. I want to point out one other thing. Okay, and then we'll come back and we'll try to put all of this together. So here's the clefts, okay? And then on the sort of inside kind of area here, okay? And just kind of on the underside as well, we're gonna have basement membrane. And we're gonna have basement membrane. I don't care about the structure of the basement membrane, you just need to know that it exists. One of the things that we have in the basement membrane is an enzyme called acetylcholinesterase. Acetylcholinesterase. The job of acetylcholinesterase is to break acetylcholine off of its receptor. Okay, so you release it. Okay, so everybody turn to your neighbor, shake their hand, hold it like this. Okay, shake your neighbor's hand. Come on, everybody. So acetylcholine has bound, it's bound to its receptor, we're transmitting a signal. Then acetylcholinesterase comes in and chops it off. Okay, and it breaks it apart. Now, no more contractions. Okay, that's how this works. If we don't break the contraction, we don't break the signal, you can't ever relax the muscle. Okay, you can't relax the muscle. There are, and you guys are going to have an assignment on this for Wednesday. There are certain kinds of snake and spider venom, as well as one of the most heinous chemical weapons the world has ever conceived of, that what they do is they block the function of acetylcholinesterase. So what happens is, if you can't turn this off, then especially in, say, your diaphragm, you can't relax your diaphragm. If you can't relax the diaphragm, then you can't take another breath. And so you're stuck, diaphragm won't relax, you can't continue to breathe, and you will die. Okay? And so you essentially get into a cramp, and it either fatigues to where it won't work, or it can't ever let go, like with your diaphragm, and you can't relax it, so you can't breathe. Okay? Anybody ever been hit? Like, you guys ever got hit, like you're right in your diaphragm, and you're like, I can't, like, you can't get it, it kind of spasms, and you can't get it to relax, and you can't breathe, and it's really freaky.
Yeah, I ended up in the hospital.
Yeah, okay, for the same kinds of things. So having this function, having this enzyme there is really, really, really important. Question? I'm not sure about anthrax. So the chemical weapon is called sarin nerve gas. It does that. And then I've forgotten if it's the black widow spider or one of the, it's like a banded crate, a couple of snakes and things, they all sort of do the same. Or if it's the cone snail, there's several things that do this. You guys will have y'all will have an assignment. We'll talk about some of these things. All right. Good deal. So we mentioned this the other day, but we're gonna hit on it again, okay? Because it's one of the most key concepts for everything that we have to do to understand neuromuscular physiology. The idea of the motor unit, okay? A motor unit is one motor neuron, its axon, and every individual muscle fiber that axon has a neuromuscular junction with, okay? This is the functional unit of the neuromuscular system. When you want to move, when you want to generate force, you do so one motor unit at a time, okay? So you will note here, right, we see two motor neurons. You will note that they're going to come out. The first one's going to connect to 3 fibers. This one, the second one, is going to connect to 1, 2, 3, 4, 5 fibers. This is how this works. Okay? Some motor neurons connect to a small number of muscle fibers. Other motor neurons connect to hundreds, thousands, tens of thousands of muscle fibers, okay? So we're gonna talk about motor units by their size, and by size, we mean the number of fibers that are connected to each individual motor neuron, okay? When you need to turn on muscle, there's a very distinct order in which this happens. There's a lot of rules about how this is going to work, okay? So we'll start with the motor unit. It's the alpha motor neuron and every fiber that it innervates, okay? So as you will see here, kind of the same concept, small unit on the left, somewhat larger unit on the right, okay? The smallest motor units in your body have maybe 10 or 20 fibers in them. They tend to be in very small muscles, okay? Very small number of fibers. You can have, in say, your quads and your glutes and your lats, you can have motor units that are connected to tens of thousands of muscle fibers. Very, very large motor units, okay? The ratio of fibers to motor neurons, so the size of the unit, and our ability to adjust which ones get turned on and when, allow us a tremendous amount of precision in how much force we're going to generate. Okay? Because remember, when a motor unit gets activated, every fiber that's connected to that motor neuron, they're all going to contract at the same time. Okay? So imagine if all you had was like 2 motor neurons that went to your quads. So I could turn on half the fibers or all of the fibers in my quad. Okay? I would walk around like real herky jerky, like running would be it's either half or it's the whole thing. There's no ability to kind of precisely control force production in between all. And so the advantage of the setup that we have, where there are hundreds or thousands of motor neurons that go to an individual muscle, and this continuum of sizes of those motor units from those motor neurons, it allows us very, very precise, very fine gradations in force, which is why we can move in the way that we can move. Okay? It's why you can write on your iPads and things without jamming your stylus into the screen. Okay, it's why people can play the violin or the cello or something with these very, very small, very precise kinds of movements. Okay, it's all because of how this is going to be set up and how it's going to work. Okay, so I'm going to talk really quickly about action potentials, and then I'm going to have you guys do an in-class, a little bit of an in-class assignment, okay? I assume you guys have all seen action potentials before, yes? Action potential is, okay, it is basically an electrical signal, okay? So let's do this, okay? Everybody in my front row, can you all stand up for me? Turn around and face the rest of the class. Can I get you to scooch in just come a little bit closer, okay? So the idea, okay, behind an action potential is we generate an electrical signal in one place. So my guy on the right, okay, what I want you to do is just jump up and down. Just jump, go. Okay, action potential. We have generated electricity right here. And the reason that we're going to do this in the nervous system is it moves really fast. So I want him to, we're going to do like a wave here. So like you jump and then everybody sort of subsequently jump after. Ready? 1, 2, 3, go. Oh, we got to wait. See, we got out of sorts. Okay. But this is the idea. Would it be easier if I was like, just like touch the shoulder of the person or like slap their hand? Are we okay touching each other's shoulders? Is that okay? Gently. Very non-creepily, okay? Ready, go. Yeah, there we go. Perfect. Thank you. Thank you. Y'all can sit down. Y'all can sit down, okay? Very good. So this is the idea. What we just showed you guys, okay, was what's called propagation. Propagation. We generate an electrical signal in one place and then it moves, okay? It starts in one place, it touches the person or the space adjacent to it. That place then generates an action potential. It touches the next, the next, the next, the next. And we end up then with a signal from this side of the room to the other side. Okay, relatively quickly. It's a little better than playing a telephone game where we give you a word and you got to whisper the word all the way down and we get over here and it's like a completely different word. Okay, that's what we're doing with action potentials. This is in the brain and the spinal cord until we get to the neuromuscular junction. Action potentials are how we send signals in the nervous system. Every thought you have, your perception of this room and me and your neighbor, everything you see, everything you smell, everything you experience is just your brain's interpretation of action potential. That's all that it is. Okay? My pants are kind of charcoal gray because your neurons fire action potentials and you interpret that as being gray. Your chairs are red because that's how it's that way. Okay, the temperature in the room is because those are the action potentials that you perceive in that area of the brain, and you interpret it that way. Okay, all of our nervous system things are just action potential. It's fucking wild. Okay, so we're going to use this because it's fast. Okay, it's fast and it's precise. I'm going to show you guys just a little bit. Things that we need to have, okay, nervous system tissue, and then the sarcolemma of skeletal muscle are special kinds of cells because they have what's called a resting membrane potential. All that a membrane potential is there's a difference in the charge from inside the cell to outside the cell. Because there's a difference in charge, we have a potential that if we let the ions that generate that charge move, that the charge will flip around. Okay? And when the charge flips, that's electricity. That's all electricity is. Okay? That's all that lightning is, or that's all that what's coming out of these electrical outlets. So we have a resting membrane potential, and we have different concentrations of ions, especially sodium and potassium. Okay? In our nervous system and in skeletal muscle of the sarcolemma, we tend to have a resting membrane potential of about minus 70 millivolts. Okay. We're more negative on the inside than we are on the outside. We also tend to have much more sodium on the outside than on the inside. Sodium has a positive 1 charge, more of it on the outside, less of it on the inside. So we have both a concentration and an electrical or a charge gradient from outside to inside of our cells. So when we want to generate electricity, we do one very simple thing. We open channels to let sodium move through. Open a sodium channel. That positive charge comes from outside to inside. It's going to move down both its concentration gradient and its electrical gradient. As those positive ions move from outside to inside, they're going to start to counteract that negative charge inside. So we're going to go from minus 70 to minus 50 to minus 20 to 0. Then it's going to overshoot and we're going to get a positive charge on the inside. And voila, that's electricity. Okay? That's what's happening. So we open these sodium channels, or what we call voltage gated sodium channels, and it lets the ions. Okay. Now, once we've done that, we have to restore membrane potential. You've gotten one action potential, but for me to talk, I have to transmit thousands of action potentials. You're going to go once, and then again, and again, and again, and again. So what we do is sodium comes in, close the sodium channels, open what are called potassium channels. We have a lot of potassium on the inside. So we open a potassium channel and potassium goes from inside to outside. So now we have a positive charge leaving the cell. It was very positive. Now it's becoming more and more negative, and that's going to restore our concentration, or it's going to restore our ionic gradient. Then we use things called ion pumps, or a thing called sodium potassium pump. That once as we begin to restore the ionic gradient, we're going to use it then to exchange and put sodium out, bring potassium back in, and restore our concentration gradients. All of this takes fractions, like microseconds, to happen. Okay, microseconds. Boom, out, back in, or sodium in, potassium out, flip the charges. Okay, over and over and over and over again. Okay, and that's how we generate action potential. So every time you think, every time we move, every time you see something, this is all of the things that's going on. Okay, so this shows sort of how we get all of this. It's going to talk about overshoot and hyperpolarization. This shows when things are gonna move around in that particular way. Relatively straightforward stuff that I know you guys have seen before. For our purposes in class, what we really want is we're going to talk about just the fact that it actually exists. Okay. We're going to do this, and then I'm going to have you guys do a little in-class activity. So, on the nervous system side, okay, action potential in the brain travels down spinal cord. Motor cortex action potential, move it down the spinal cord. Okay. It then reaches the alpha motor neuron in the spinal cord. It generates an action potential. It's grabbed by the dendrites, and it says action potential in the alpha motor neuron. That then moves down the axon. Node of VA to node of VA to node of VA. We then reach the neuromuscular junction. Okay. That action potential creates a charge. It opens these calcium channels. Calcium moves into the glutathione. Acetylcholine goes into the primary and secondary neuromuscular cleft. They bind onto the acetylcholine receptor. And then a funny thing happens. We went from electrical signal to a physical neurotransmitter signal. And then on the muscle, we're going to go back to electrical signal. When you activate the acetylcholine receptor, it then generates an action potential on the sarcolemma. And that action potential then spreads out along the sarcolemma. And it spreads and it goes down the T-tubules, and then it gets brought into the inside around those myofibrils, and we're going to get calcium release, so on and so forth. Okay? And that's the process of how we go from the brain all the way down to getting actin and myosin. Okay? So here's what I want you guys to do. We got about 15 minutes. Y'all are going to do this as a group. You can do it by yourself if you're like, I don't like people. I totally get that. All right. Or if you want to do it with a couple of other people, that's fine. But you're going to turn in one piece of paper or one file. If you want to type it out, it doesn't matter. You can email it to you. You're going to turn in one thing. So just make sure you get everybody's name on it. Okay. I want you guys to write down every step, every signal from brain all the way down through the cross bridge cycle, where we get a cross bridge made and then broken. Every step, I want you to include every structure, and I want you to include every signaling molecule as we go from structure to structure that you can think of. Okay? You got 15 minutes. Go. For my folks on Zoom, I'm going to put you guys into a chat room, and I want you guys to do it and do the same thing, and y'all can just email it to me. So breakout rooms coming.